I'm delighted to introduce our last speaker of the day, Dr. David Hong. He is an assistant professor in the Department of Investigational Cancer Therapies at the MD Anderson Cancer Center. He is also clinical medical director of the Clinical Center for Targeted Therapy. Dr. Hong will speak to us today about an area of significant interest to GIST patients, clinical trials, and new drugs. Dr. Hong's research interests include early drug development for targeted therapy in cancer, new combinational phase 1 studies, and novel protocol designs. Please welcome Dr. Hong.
Thank you very much. I'm really grateful for John and for inviting me here, and I'm grateful for Barbara for having the patience. My wife was due to have her second child this Tuesday, but the baby has not yet come. And so I kept on telling Barbara, I don't know if I'm going to be able to make it. I don't know if I'm going to be able to make it. And she was like, okay. And but my wife has not gone into labor, even though we've walked to Dallas and back, and she's still at home and doing well. Well, I'm really honored to come here today and talk to you folks. And you know, for me, talking to patients is probably not only an honor but also just a privilege because you know, this is why we do the research that we do, and that's why I'm in the job that I do, is because of you and the disease that you guys are struggling through. Today I'm going to talk, the title of my talk today is called The Good, the Bad, and the Ugly. And I don't know if you guys remember these spaghetti westerns, but you know, every Saturday afternoon, I used to sit down, not every Saturday, but a lot of Saturday afternoons I would sit down with my dad, and he used to love these spaghetti westerns. And Clint Eastwood obviously was the star of these spaghetti westerns. And if you guys recall, this is one of the most famous spaghetti westerns that he started, which was The Good, the Bad, and the Ugly. And so today I'm going to talk a little bit about the bad and the ugly of drug development in the United States and some of the obstacles that we face, and but also share about some of the good news that is arising from that. And I'm going to share, and John had asked me to specifically talk about ongoing studies that we have in our phase 1 department with specifically targets and pathways that may be applicable to GIST patients. So I'm going to talk about that also. So as you all know, cancer is a problem, and it's increasingly becoming a worldwide problem. Many of you guys probably already know that in 2012, cancer will overcome all known diseases as the #1 killer of people in the world. And so it's increasingly becoming not just a US phenomenon, but also a worldwide phenomenon. And every 57 seconds, another American dies of cancer, 63 over the past hour. And again, you know, all of us are going to be affected. If you're already family, obviously you're affected by it because you have a loved one, or if you're a patient, you're affected by it. But the reality of it is that almost 1 in 2 males in the United States in their lifetime will be stricken with cancer, and 1 in 3 females will be stricken with cancer. The good news is that, as you all know, that we are starting to really kind of peel back this black box, and over 20, 30 years, many of kind of the pillars of oncology now set forth kind of this underlying understanding of molecular biology, and we are beginning to start to reap some of the fruits and the benefits of that in actual molecules that target these different pathways. And so it's led to a number of different therapies. And has led to, which I know you've guys had a talk today about mutation analysis and all that, where we are now really transforming not only oncology but medicine, where we will truly move towards a personalized approach towards cancer and cancer patients. And not only will that help us predict patients who may respond to therapy, but it may also help us to predict patients who have either poor response or may be toxic to certain types of cancer therapies. So the problem, in some ways, is not the science. The problem is really kind of the regulatory framework we have here in the United States. And I don't know if you guys remember this, 2 years ago, my son was born in September, and then Hurricane Ike hit, right? And we had to move from, my wife was, we all moved, I was actually at a family reunion in Wisconsin, and my wife had to drive from Houston up to North Houston because of Hurricane Ike, and she was telling me how many hours it took. It must have taken, I think, which is a 30-minute drive. Usually it took her 10 hours to drive to North Houston. And this was a picture of that traffic jam on the way to North Houston. Everybody was leaving the city at that time. And so in some ways, it's this is very emblematic and symbolic of what's happening right now in the context of drug development. We have now hundreds of new drugs that are entering the pipeline, which is very good news. But the problem is that this highway is starting to get jammed, partly because of a number of different regulatory factors, and also factors in how we design clinical trials, and other factors, including, unfortunately, these are just a small list of the different agencies that are involved in clinical trials. So everybody has a hand in it, and everybody has a different regulation, which is unfortunately increasingly making clinical trials more difficult, but also slowing down that process. And so oftentimes it leads to delays and costs and frustrations and decreased efficiencies. And unfortunately, here in the United States, I don't know if there was an interesting article in Science recently about the shift of clinical trials now moving overseas. Pharmaceutical companies, just like any other companies, are global companies, and they will move towards where they can do things faster, cheaper, and more efficiently. And so, unfortunately, because of some of these regulatory hurdles, many of these trials are moving to places like India and China and Russia and Eastern Europe. And that's a trend that I'm not entirely sure we're going to be able to stop anytime soon. On average, this was a paper done by he is a health economist and has looked at a number of different factors, but this is, on average, the number of years it takes for an oncology drug to be approved, which is about 9.1 years. And that, I know for many of you, is not fast enough, right? And so even with the orphan drug approval process, it's not much faster. I think it's around 7 years is the time from first clinical trials to actual approval of a drug. So we have to change things. And some of this has to deal with the fact that scientists and physicians themselves are kind of a problem in themselves. We often have grant systems that often lead to research that are really not relevant. Some would argue this was a series of articles published in the New York Times. I'm sure many of you have read these articles about some of the hurdles in cancer research. And that's one problem. Another problem is the fact that in general, I know probably not this audience, but in general, patients do not enroll in clinical research. And that becomes a problem because the slower the accrual of clinical research, the longer it takes. And this was a very interesting article by Amy Harmon. This was actually last week. And it was an article about two cousins who unfortunately developed metastatic melanoma, and they were trying to enter a clinical trial that was actually developed here, a drug that was developed here at MD Anderson called PLX0492. And in the early trials here and other institutes, we found that there were some incredible response rates in patients who had BRAF positive mutations, similar to GIST and KIT mutations, that led to almost an 80% response rate. However, this article pointed out that the development of this drug has somewhat been delayed because the FDA is requiring a large randomized phase 3 study with a placebo. And so this has stirred debate also within the oncology community and obviously with patients. And one of these cousins was randomized to a placebo. Obviously, it was placebo, and this article talks about the story about that and just the challenges of that and whether that is truly ethical or not in this era of targeted therapy. I want to talk a little bit about the phase 1 group here and our mission. I finished fellowship about 5 years ago, and MD Anderson actually did not have a phase 1 group or a targeted center group. MD Anderson is a very big and wonderful place, but it's often divided into these kind of individual fiefdoms, the kingdom of GIST by Dr. John Trent, and the kingdom of lung cancer or breast cancer, et cetera. And so most of these early phase 1 trials are done across disease types, and so there was no specific home for it. And so about 5 years ago, Dr. Roselle Kurzrock, my current chairman, and Dr. Wong-Ki Hong, who's the division chair, decided to create a specific center for targeted and early first-in-human drug therapies. We have now grown to be the largest phase 1 unit in the world. We have close to 120 different trials. And I'm going to talk a little bit about some of those trials today. But we began again in July 2004, and we have now become a department. We have approximately 8 faculty on board. The idea of it is that we are not really disease-based, but really target-based, and we are moving towards that more and more. In fact, one of the objectives, and I'm going to talk a little bit more in depth about, is we're profiling almost every patient who comes to our clinic. We're looking at a number of different mutations that they have, not only KIT, but things like PI3 kinase, PTEN loss, c-MET amplification, KRAS, NRAS mutations, et cetera, so that we have some profile of what is going on with these patients and maybe even able to assign specific drugs that are targeted to those mutations in our trials. Again, these treatments are based on early phase 1 trials. Most of these studies that we have ongoing are phase 1 trials, and many of you here are familiar with the drug development process. There is what's called a phase 1, phase 2, and phase 3 clinical trials. Usually phase 3 clinical trials are really at the end part of development. Phase 1 studies are oftentimes the first in human trials. Their primary purpose is really to figure out what is the safest dose in humans. But increasingly with these targeted therapies, we are using them as proof of principle to discover whether or not there's activity in patients who have specific abnormalities or molecular abnormalities. Treatment is not conventional. Virtually all our patients in our clinic are on a clinical trial. And we often do combination studies that are not done anywhere else. Combination of 2 drugs, for example, like Sutent, which is, you guys are all familiar with valproic acid, which is an HDAC inhibitor. Combinations that are not conventionally done in other centers. I want to show you some evidence that this idea of targeted therapy actually works. This is a young woman with a disease, a lymphoma-type disease called Castleman's disease. And we know from the basic biology of it that it is driven by a cytokine called IL-6. It turns out we had an anti-IL-6 monoclonal antibody. And we've now put, I think, close to 20 patients on the study. This is an old slide. But almost 20 of those patients all respond to this therapy. At this time, 12 out of 14 patients with Castleman's on study had responded. And they've moved this molecule now from a phase 1 even to a registration phase 3. Likewise, another drug that has moved very quickly from our clinic targets a very rare thyroid cancer called medullary thyroid cancer that often have what are called RET mutations. This drug, XL184, we discovered actually specifically target this RET kinase, and it's a patient who has significantly benefited from this. And we now know that almost 70% of these patients who actually get this drug and have the RET mutation respond to this therapy. And this drug also is moving towards a rapid registration phase 3, hopefully shortening that time of approval from 9 years down to maybe even much shorter than that. You may have heard of IGF-1R inhibitors because it's relevant to just tumors, but the activity of this drug has actually shown some remarkable activity in another sarcoma called Ewing sarcoma, and we were one of the first groups to describe that. And we had an IGF-1R inhibitor, a young woman who came in, was refractory to multiple chemotherapies, who got this drug and had a near complete response, and she, I think, remains disease-free to this day. So we are moving towards moving towards the idea of histology-based, histology-independent target-based studies. The idea that cancer is a disease of genetic aberrations. And if we know those genetic aberrations and we can target those genetic aberrations, it doesn't really matter what your histology is. It may actually it's more important what those genetic aberrations are. We have a number of now ongoing BRAF inhibitor studies. I mentioned to you about that, those cousins that had the melanoma BRAF mutations. It turns out other cancers also have BRAF mutations, including thyroid and colon. The same case, MEK, which is downstream of BRAF, those patients who have BRAF mutations appear to also respond remarkably to MEK inhibitors. PI3 kinase and mTOR inhibitors, I'm going to talk a little bit more in detail about that. But there is subsets of cancers, endometrial, ovarian, colorectal, that have these PI3 kinase mutations. And the data I'm going to show you should suggest that if you have that mutation, and if you get a drug that targets that pathway, you may seriously clinically benefit from it. And we are doing unique combinations. I'm sure Dr. Trent has shared with you the point that, that targeting one pathway may not always be the best, that sometimes resistance is a consequence of multiple pathways that are being upregulated. And so we are trying to do early on these combination studies where we're targeting multiple different pathways. One of our ongoing studies is a study called PREDICT. As I shared with you, when I see a patient in my clinic, one of the first things that we're trying to do is with the existing paraffin tissue that they have, try to send off a number of different mutational analysis. And we are following those patients, and hopefully sometimes right now the actual mutation analysis does not come back sometimes quick enough for us to actually assign a patient, but increasingly that is speeding up. And even though they may be on one study that necessarily was not targeted, about a month later they may come back, and then once we know their mutations, we're able to, if they fail that study, may be able to assign the next study to them, a study that may be specifically targeted. And so one of the studies that we are conducting and looking at this is what's called the PREDICT study. And I'm going to show you some examples. This PI3-AKT-mTOR pathway we talked about, from my understanding in GIST, I'm not a GIST expert, but John has shared with me that this may be a pathway of resistance in some GIST patients. We have looked at a number of patients, in fact, close to 500 patients in our clinic, and of those patients, about 34 of those patients had PI3 kinase mutations. And if you look on this side of this is what's called the waterfall graph, and this is the lower the bars go, that means the more that they've responded to therapy according to the CAT scans. And the higher it goes, that means that they've progressed or increased in size. And as you can see, those patients who actually have what are called PI3 kinase mutations have had significant responses. There are some who do, but most of them have concomitant KRAS and NRAS mutations. And that's something that has been borne out in lab experiments by doctors like Garth Powis here in the experimental therapeutics that show that patients who also have KRAS mutations may not be amenable to drugs that target the PI3 kinase pathway. This is another example. These are BRAF positive patients, again, who actually received a drug that targeted the BRAF mutation or received a MEK inhibitor that targets a molecule downstream. And as you can see, they've had some remarkable responses in this. There are some subset of patients that are resistant, and we're not entirely sure why at this point. But if you look at their survival, if you look at those patients that actually had BRAF mutation and actually received a BRAF inhibitor, their survival is significantly improved relative to those patients who have not received that. And so increasingly, I think that is an important point, that the more we know molecularly about your tumor, the better that we have an idea of what may work and what may not. I'm going to end today with just a list of some of the protocols that we have ongoing. John had sent me a list of pathways that you may be interested in. I think these slides will be available to you all. These are all either enrolling or pending studies in our phase 1 clinic. We have a number, actually 4 different trials that target the aurora kinase pathway. Many of them are also dual VEGFR and also c-KIT inhibitors. We have a number of c-KIT inhibitors such as sorafenib, dasatinib, and Sutent in combination, however, with other agents, hopefully to add synergy and more, I guess, bang for your buck in a way, with HDAC inhibitors such as valproic acid. We have this protocol that combines both valproic acid and sorafenib, valproic acid and Sutent. We have a number of also MEK inhibitors or combination trials that target both MEK and BRAF. I showed you that slide of those patients who actually have BRAF positive mutations. I don't know, John maybe can explain whether or not GIST patients actually develop BRAF mutations, but this pathway is also, we have many trials targeting this pathway. PIK3CA is also a very hot pathway that people have been looking at, and there are a number of agents right now in phase 1 study, the GSK molecule and also PX866, which was actually a molecule developed here at MD Anderson, is up and running. We have a number of HDAC inhibitors, including a unique HDAC inhibitor called CUDC-101. And a number of mTOR inhibitors. mTOR is also a molecule that is downstream, actually, of PI3-AKT. And so sirolimus, which is an interesting drug, it's an actual initially drug used in transplant medicine, but was discovered to target this pathway. And also temsirolimus and everolimus, which are also drugs that target this pathway. We are using those in combination with multiple different other targeted agents and also chemotherapy. There are 2 very interesting studies. These studies will be my studies and will likely open in April of 2011. One is a new class of IGFR, a second second class of IGFR inhibitors that is coming out. It has a dual role. It may also inhibit angiogenesis through a unique pathway called angiopoietin or Ang2. Lastly, some of you probably are very aware of HSP90 inhibitors. Some of these agents, HSP90 essentially is a protein that helps client proteins like c-KIT signal, right? So the idea and the thought is that if you block HSP90, you're going to also block c-KIT, or the idea may be that patients who may become resistant to c-KIT inhibitors, if we also block HSP90, we may be able to overcome that resistance. So there are several HSP90 inhibitors out there. Several of them have shown at least some benefit in the context of PET responses in GIST patients. However, one complication of these HSP90 inhibitors, one is a drug called IPI-504. Recently, there was a large phase 3 study done of that study, and there was an increased number of patients who died from liver toxicity. And so one fear is that HSP90 inhibitors may be more toxic than we anticipated. However, BIB021 so far has not shown that liver toxicity, and so there will be an ongoing study of a combination of BIB21 plus imatinib in the near future, and that's something that I know Dr. Trent is looking forward to. So I want to end with a quote. This is one of my favorite Bible verses. It's from Romans 5:3-5. It says, "Not only so, but we also rejoice in our sufferings, because we know that the suffering produces perseverance. Perseverance, character, and character, hope. And hope does not disappoint." If you asked me 5 years ago as a fellow if we could cure metastatic cancer, I probably would have told you, not in my life. But even within 5 years, I have seen a revolution in the drugs that are out there and the science that is out there. And I really believe that in my career, maybe within the decade, that we are going to be able to cure patients with metastatic disease. And so I want to leave you with that sense of hope. There are an incredible number of new drugs out there. There are an incredible new number of exciting things out there. And thank you for your time.
Questions? That was awesome.
It's on talk. One question I had is, there are a number of NCCN cancer centers around the country, and it sounds like perhaps you have this model of a phase 1 group that's far in advance of most centers, or in size at least. And my question is, can you envision that someone such as myself maybe going to Roswell Park in Buffalo, where I have tissue, but that would be able to get my tissue to you and the benefits of a center this size without actually physically coming here as a patient and doing all of that, but getting the benefits of the expertise that's here?
So are you asking whether or not, maybe getting the mutation analysis done here and then also maybe enrolling in that clinical trial there?
Well, I understand that enrolling in the trials is one thing, but more so that you're talking about the lists of genetic mutations that you'd be testing for and having a profile developed and things.
So I think that's not far from the reach of most of these NCCN cancer centers. I mean, I'll give you an example. I think in 2002, it cost close to $2 billion to sequence the whole human genome. In 2010, there are companies now that can sequence the whole genome for, I think, $7,000–8,000. And the prediction is that within maybe another 2-3 years, that we'll be able to sequence the whole genome for about $1,000, which is not much more than how much getting a c-KIT mutation analysis is currently. And there are a number of companies out there right now that are able to are just rapidly ramping up to start doing that, and that will, I think, more than anything, will start to revolutionize medicine, because your doctor at Roswell Park will be able to send that sample off to Company X, and he will do a huge profile of it. And then he may be able to say, well, gosh, beyond your c-KIT mutation, you actually have a number of other, you may have a BRAF mutation of V600E. You may want to try this drug, It may be off-label at that point, but you may want to give this drug a trial. And so I anticipate that's going to happen very quickly in the next, and John does more lab research than I do. I don't do any lab research, I just do it. But this is becoming an increasing reality.
Yeah, there are companies now already that are doing this type of target identification, where you send them tissue from the tumor, and they will send you back a report of proteins that are expressed in your tumor and a relative level. Whether or not that is going to be useful clinically, we don't know yet. Yeah. But I think the technology is out there already in terms of protein expression, as far as the in-depth sequencing and mining for activating mutations. That's probably a little bit farther on the horizon, but would be a huge advance.
We'd like to thank your wife and your new baby to wait so that you could join us today. Thank you. That was most informative. I've heard that cancer is called the disease of the aged, meaning over age 50. Is there an increase in incident rates in younger people? Is it something that's part of our North American lifestyle and diet? Is it increasing, decreasing? Is it spreading amongst a number of different age groups now?
Well, I think a lot of cancers are decreasing with the, for instance, lung cancer is on the way down because fewer people are smoking. In countries where smoking is still prevalent, it's still increasing, and it hasn't hit that curve yet. But in the U.S., smoking went down drastically 10 or 15 years ago, and now we're seeing a decrease in the risk of lung cancer. We're also seeing decreases in cervical cancer and some of these cancers that are caused by known environmental hazards. Melanoma, more people are staying out of the sun more and are using sunscreen and taking caution there. So that's, you can decrease cancers that way. Sarcomas, that's clearly not the case, because the majority are not caused by an environmental hazard or an environmental agent. There are a few examples where they are, like angiosarcomas and osteosarcomas can arise in fields where patients have received radiation therapy. There are a few examples in other types of sarcomas. In GI stromal tumors specifically, there aren't really any known environmental hazards that people can avoid to decrease the occurrence of GIST. So it's does not seem to be going down in incidence nor prevalence. And it's compounded by the fact that the diagnosis by pathologists like Dr. Lazar and Wong, who are here today, they've really changed the way they're diagnosing it. And there are things that they didn't call GIST 15 years ago that they're calling GIST today. So the incidence of GIST has really gone up by about 10-fold, but that's probably because it's shifted from other populations. So in GIST specifically, it's a bit of a moving target to know whether or not it's increasing or decreasing in incidence, but it's not thought to be drastically decreasing in incidence. And I think part of that ties into the fact that we don't know what the specific causes are for most cases.
I think the biggest demographic change is really the developing world. So as in the developing world, 20 years ago, the most common cause of death was infectious disease. But as the developing world develops and they develop better hygiene, better and worse diet, I guess, Western diet, Western habits such as smoking and so forth, increasingly the developing world like China and India are developing cancer rates that are equivalent to the United States. And I think that's where you're going to see the shift in cancer. I mean, for example, in China, close to 60 or 70% of people there have hepatitis or have been exposed to the hepatitis virus. And if you think about it, about maybe 5% of those people who are exposed to hepatitis will eventually develop liver cancer. If you think of the billions of Chinese people who have been exposed to hepatitis, and just that 5%, the number of patients who will be out there who have liver cancer is going to be huge. So I think it's an issue as much as I think part of it also is issue of diagnosis. Maybe several years ago, I think we did not have the tools to diagnose cancer as we did now, and that may be why you may be seeing a spike in what is diagnosed as cancer in earlier patients. Back then, they may not have known what it was, and people died of causes they didn't know what they were. But I think that the major demographic shift you're going to see in the next decade is the fact that the developing world is going to develop cancer.
You talked about some of the clinical trials going overseas, India, China, Russia. Do you trust their results?
That's a good question, and that's been a debate in the clinical trial arena. The FDA does accept clinical trials that are done in other places like China and India and in Europe, as long as it's been audited and rigorously reviewed. And so that's, from the FDA's point of view, as long as the data can be trusted from their perspective, it's been audited. All our trials are audited by what are called CROs, clinical research organizations. So they're independent organizations of the drug companies, et cetera, that come in and look at the data and audit the data to make sure that the data is correct. And that's part of the reason that's the additional cost of running clinical trials, right? It's not just putting a drug in person X. There are layers of regulatory costs on top of it. And even despite that, in places like China and India, the cost is probably 1/10 of what it is here in the United States. Even though they may even pay, here we have something called, if a drug company comes to us, the drug company really only pays for, like, for example, they pay for the drug, they provide the drug, they pay for things like research costs, like research biopsies, research bloods. They may pay enough money to pay for the research nurse. That's all they pay. Yet in other countries like Russia, they will pay for the whole care. For example, they'll pay for CAT scans, they'll pay for everything that's what's called deemed standard of care here in the United States, which is paid for by the insurance companies. Even then, the cost of doing a clinical trial in those countries is about 1/10 of what it is here. So there's an interesting article in Science, I forget who the author was, he's from Vanderbilt, and he talks about how he compares our current clinical trial mechanism in many ways like GM, that we are in a way competing with, in the United States, with other countries that are developing these new models of clinical trial and cheaper and faster and more efficient and maybe even with better quality. And so I hope that right now, there was an article 2 years ago in Nature that looked at all oncology clinical trials throughout the world, and we still have about 70% of those ongoing clinical trials here. But if you looked at the trends over the last 10 years, we've had a decrease of about 10%, while every place like China, India has all had an increase of about 20 or 30%. So over time, if that trend does not change, the number of clinical trials in the United States that are run by drug companies will probably continue to decline.
This is very interesting, and again, I'm very grateful to be here. I did watch the Stand Up to Cancer telethon a few weeks ago and noticed that, part of the promotion was the uniqueness of advocating collaboration and getting trials from the lab to patients much quicker. Would one of you care to comment on that and your thoughts on that and its relevance?
Well, I think part of the struggle that we deal with in getting clinical trial to getting a drug approved, from somebody making the drug to testing it in vitro, to testing it in cell lines, to testing it in animals, to first in trials like Dr. Hong does, the phase 1 studies, to the phase 2 studies like we do with just patients in our sarcoma center, to larger multi-institutional phase 3 studies where drugs get approved by the FDA. That whole process takes 9 years, 10 years, sometimes longer, and it literally costs hundreds and hundreds of millions of dollars. So there's a time thing that we're fighting, there's the cost that we're fighting, and to speed that up, it's just going to take a lot of work at getting through the bureaucracy. And the regulatory rules are important for safety, but at the same time they add cost and they delay the time to approval of these drugs. So, so we have to just keep working at doing trials faster and more efficiently, and getting drugs to patients more quickly.
As I kind of pointed out, that this problem of transitioning drugs from the lab to the clinic is a multifactorial problem. As John pointed out, it is costs. If you look at health economists who have studied this process of developing an oncology drug from the lab to approval, on average, it's about $1 billion. Some people argue it's less than that, but it's like 700 million to $1 billion. Once you get over $500 million, it's a lot of money, right? And so institutions like MD Anderson, even though we may develop a drug here, we cannot develop it on our own. We actually have to license it out to a drug company. So we have to say, we have a drug we developed in the clinic here, and we've done excellent experiments. Do you want to buy this drug, or do you want to give us some rights, royalty rights to it? But MD Anderson, even though as big of an institution it is, there's no way they have enough money to develop their own drug. But with that said, I want to be encouraging, because there have been more drugs now in the pipeline that are in phase 1. Many of our drugs are combination studies, but we have 120 studies. I don't think John would have, I would have ever imagined we'd have that many studies in one center. But and I see almost every day a drug company who comes to us and says, we would like to try new drug X, And so what I want to encourage you all is that there are new drugs coming, and they're coming, but they're not necessarily coming as fast as we'd like, but there are many new drugs that are coming out there. And so I'm excited, and I think that we're going to see more and more responses. We're going to see more and more patients who are benefiting from these new drugs.
Wait for the microphone. On drugs like these that to us are life and death drugs, what happens when generics come along?
That's a complicated question, and there's not a good answer to it. Drugs come off patent and generics become available, and usually the generics are of equivalent quality. There are some cases where they have not been, and there have been some error with the generic. It's going to come down to choice, an individual choice. You can choose the generic or you can choose the name brand. And there's also going to be better drugs coming along. And so it's going to be a complicated issue when generics are available. We don't know, what their pricing is going to be. So.
Well, you know what will happen with the insurance. Duh. Always wondered.
Yeah. So there was an interesting legislation. I know a lot of people have different opinions about the new healthcare reform, but it turns out one of the reasons that the drug companies didn't have a big hoopla about drug reform this round is that they cut a deal, and they cut a deal with I think the administration and also the Democratic Congress, and that is that if they wouldn't cause too much of a stir, they were going to be extended a patent life. So there's a shelf life beyond the patent life, which was passed recently, that I think extends it now to 12 years beyond the patent life. So a patent life of a drug is about 20 years. It's from the time that it's submitted to the patent, but it still takes 9 years, right, for a drug to be developed, right? But there's also a life. There's an extension of that exempts it from generics beyond the point at which it was approved, and the drug companies essentially negotiated, I think, with the administration and Congress, so that shelf life will be 12 years beyond the point it is approved. There was no shelf life for biologics, such as antibodies, et cetera, until this past year, and then that's Congress had threatened to shorten that, which was now currently 7 years, down to like 5 years. But essentially the drug companies negotiated with them, and so it is now 12 years for all drugs.
Do generic drugs go through the same ritual and trials?
No, they don't. The only requirement of generic drugs is that they meet the same structure as the drug. So that they have the same essential chemical structure. Now, a drug is far more complex than just the chemical structure at times. You have to, like a pill, for example, imatinib, there's a certain bonding you have to do around that molecule, how you produce it and so forth. So there may be differences, and people argue that generics are not necessarily the same, and there may be some truth to that. But essentially, the FDA laws is that the generic just has to meet this chemical structure, and that they prove that the chemical structure meets some certain guidelines. They don't have to do any clinical trials or et cetera.
I want to follow up on that one, because on the listserv we get people asking questions about the Indian versions of Gleevec. They call it Imatib or VNAT or something like that. It's not available in the United States, but we have a number of people, obviously, that belong to GSI that live out of the country, and they get this for quite, from what I gather, quite a bit less expensive. And so the people in the United States say, well, how about if we get somebody to send it to us, or if we go across and, whatever, and bring it back? How do we know anything about those specifically to know whether they are the same or they're not the same? Or has anybody done anything in the United States to test them?
No. The U.S. has no official or unofficial ability to regulate a drug that's made in another country like that. So it's not, there are no safety for the generic Gleevex. The processing plant that makes the pill in the United States, then in the pharmaceutical, makes any pharmaceutical in the United States, the plant that makes the pill has to follow certain guidelines. There have to be guidelines of safety to make sure that it's not being made in a dangerous way. So it has to have certain GMP, I think it's called, practices where the pills are manufactured in a sterile environment. There's no contaminating bacteria or anything like that getting into the pills in the manufacturing process. And all of that is strictly regulated in the United States when the pill is made here. If the pill is made in India, we have no idea of what the safety is. We have no idea, and it may be perfectly fine. It may be a very safe processing plant that's doing a great job and making a great product that is safe and effective. But my biggest concern is that we have no standard. We don't know whether or not the safety of the manufacturing process is being held up to the standards of the United States. And we don't know, and as like Dr. Hong was saying, we don't know if it's the same structure that they're making or not. There's no way for us to know any of those answers, whether it's effective or whether it's safe. Whether it's just a placebo, I don't know. So in my opinion, there's some risk involved in taking a generic through that type of process. It may be perfectly fine drug that's perfectly effective. I have no way of knowing, because I don't know what standards their manufacturing is being held to.
Well, has any of the international doctor, has there been any international community? Has anybody checked that out or done any research to compare the three of them to know whether?
Not to my knowledge.
Even if our, European or whatever people are taking that, whether they're getting something that's similar or not? No?
Yes. When you were going back to this this cost difference between overseas and back, has anybody put a reasonable handle on the effects of our tort laws on that cost differential?
I don't think so. I mean, I think you guys, I think the previous questioner and the current questioner bring up a very important question about, you know, I'm sure you guys deal with this on a daily basis, the cost of these drugs, right? And even though you may have very good insurance, there's lots of copay and so forth. And especially as we move forward, you know, as we're trying to do these combinations of these new targeted agents, you know, how much is it going to cost that? If imatinib or, like, for example, you know, Avastin costs $5,000 a month, you know, if you're going to add that with another drug, how much is that going to cost? And that's a very, I think that's a very valid point and argument. And people, I don't know, you know, I don't know how to solve that situation. I think what's going to end up happening is that that is a major hurdle that's going to impact drug development down the line, is just the actual costs of these drugs to consumers and to patients. And unless you know, drug companies are willing to step back and say, look, you know, we don't necessarily need to make this much profit, I think they're gonna find that it's gonna become increasingly, if somebody's gonna, probably the government is gonna set those standards, which occurs in other countries like Britain. For example, you can't, if you're a renal cell cancer patient in Britain, you can't get Sutent, or that was the case for a while. And so there's gonna, I think what's gonna happen is that if these costs are not somehow controlled or they slow down, that's gonna deter the development of these drugs or a combination of these drugs down the line. But I don't know how to solve that situation. I mean,
This ties back into the other issue, which is the cost of doing a clinical trial.
Yeah.
So if a clinical trial costs a pharmaceutical company $1 billion to do, they've got to be able to make that money back when the drug comes to market, and then they end up selling that drug. And you know, as expensive as some drugs are, you know, it's hard to make $1 billion. And then you also have to think about all the drugs that they've developed that do not make it into clinical trial. So they may spend $1 billion developing a drug that ends up not being as effective as we thought it was going to be, and it doesn't get approved by the FDA. Well, the pharmaceutical company has already invested $1 billion, and they're not going to get a penny back on a drug that doesn't make it to market. So part of the cost that's driving the cost of prescription drugs is also the cost that it takes to do clinical trials, and all of the regulatory hurdles that we have to jump through. We have to hire people to help us with clinical trials who can help with the regulatory aspects of the clinical trial, because there's no way that somebody with an MD can figure it out. It's just a very complicated process and a full-time job for people in our clinical department. So the trials, you know, if we can make the trials cheaper, then the cost of drugs will come down also.
I think that's a valid point. I think, and that's why drug companies are moving to other countries now, because the costs of developing these drugs here in the United States are so expensive, and it's tied into a very complex debate that we're in, our country's in the midst of, right? The cost of healthcare, and I don't know if we can necessarily, and clinical trial development is all tied into that, and how we solve that, I'm not entirely sure. But definitely, if we, you know, from our point of view as physicians, I think the best way to try to help in that process is to develop better studies, develop better drugs, to develop better trials that will speed the process along, that will reduce the costs for drug companies, But I don't know how to solve that larger issue of healthcare costs, which are really tied into clinical trials.
Marina, what is your comment from a patient advocacy perspective? We have spoken on the subject. I just want to get your.
I don't really know, to be honest.
Okay.
I have a question regarding the Kansas frequency machine that's being tested here at MD Anderson as a cure for cancer. Could you give us an update on that testing, and is that a viable option for a GIST patient in the future?
What is it? What
Sorry,
is it?
what
Kansas frequency machine.
Is this the machine that was featured in 60 Minutes?
Yes.
By Dr.
yeah,
Curley.
yeah.
So Dr. Steve Curley is you know, Dr. Trent knows him and I know him, and I've spoken to him several times on this because I get calls, random calls from people about this 60-minute episode, and it is far from human clinical trials at this point. I'm not entirely sure. It's not a drug. It's actually I think a particle of some sort. Radio frequency ablation with nanoparticles,
Yes.
right? And that goes through a different regulatory process than drugs do. And so, as I've spoken with Dr. Curley about this, he says that he doesn't think it's going to be ready for human clinical trials for at least another 5 years.
All right, thank you.
Is there a lack of patient participation in clinical trials that makes the time be longer?
Yes, on the national level, not here at MD Anderson. My problem in my clinic is not that I don't have enough patients who want to participate. I just don't have, even with 117 different studies, I don't have enough trials. So patients at MD Anderson are incredibly generous with their time and their willingness to participate. But you know, most cancer patients in the United States are treated by local oncologists. And my guess is even with GIST patients are probably treated by local oncologists. And there's, beyond the willingness of a patient to participate in a clinical trial, there are also incentives. So there's really very little incentive for a oncologist to put a patient on a clinical trial when he makes money off of prescribing imatinib, right? There is no incentive, there's no financial incentive. NCI trials, for example, if you are to run an NCI clinical trial in your private practice, which some people do, usually is a money loser. So if they participate in clinical trials, it may actually impact their, I mean, it may be a means to get patients into their clinic and say that, you know, we run clinical trials, et cetera. But overall, from my understanding, it actually may make them lose money. And part of the reason I didn't go into private practice is that you have to be a good businessman in private practice, right? And you have to be good with money. And trust me, if you're going to survive in community oncology, you have to be good with money. And that's part of the, I think, the problem is that it's not a patient willingness, but there's no incentives for the vast majority of community oncologists to try to get patients on clinical trials. Now, the opposite has occurred in pediatrics, because most children who get cancer are seen at cancer centers. There's no such thing as a community pediatric oncologist. They're mostly seen at academic cancer centers, and therefore they're always most of them are, 90% of them are put on a clinical trial. So part of, you know, as to how we change clinical trials in the future is either to, you know, in adult oncology, is to try to incentivize, I think, community oncologists to put patients on clinical trials, and I don't know how we could do that. or have more patients come to centers of excellence or clinical trial academic centers like MD Anderson.
Thank you very much, Dr. Hong. I think we better call it a day now because it's getting pretty late. And I've just got a couple of things to say before we actually close. First of all, please would you fill in your survey forms? It's really important to us that we have feedback for all the reasons you've been given already, especially that we can improve next year. In a couple of weeks' time, 2-3 weeks' time, we'll have the videos available online at various places, including our website. And if you happen to be a member of GSI, you'll be told of that at the time. Otherwise, just keep checking at the website and you'll see it on the front page. And finally, I'd just like to say thank you once again to Sharon Trent and the family, to my husband, who's done so much today. To our speakers who've taken so much time and trouble to give such excellent presentations, that so many people have said what fantastic quality tools they are and how carefully they've been tailored to us and how much we appreciate it. So we'd like to thank you all very much. Thank you.
I'd just like to say a couple of words. I'd like to first, once again, thank GSI, thank Barbara, Marina, Ginger, and everybody else in GSI who has made this meeting possible. And I'd also like to thank all of the participants here who have traveled, some cases, very long ways. And I know that some of you are not feeling very well, yet you still traveled and came to the meeting. So I'd also like to give my heartfelt thanks for your interest in your own disease. And I'd also like to thank you for asking so many difficult questions today. And this is the biggest and the, I think, best GISTS meeting so far. This is our third one, and I'm also looking forward to the fourth one. So make sure you give some very good recommendations about what you want to hear about, because the purpose of this meeting is to get information that you want to hear about. So let us know what you want to hear about, and we will get some world expert like Dr. Hong or Dr. Kim or Dr. Gupta you've heard from today, and have them talk about the topic you want to hear about. And it's a rapidly changing field, so there's always new information, and just stay involved. So I'd like to again thank you and have a safe trip home.